Okay, one more pathway. That I would like for us to talk about. This one is a cycle. So it's a pathway that comes back upon itself. Oops, can you see what I wrote there? All right, so the tricarboxylic pathway, you already know it as the citric acid cycle. Or the Krebs cycle. But for molecular or microbiologists, they tend to call it the tricarboxylic acid cycle. Different names for these different things because they need to be more specific. Now, this is going to be, I'm going to draw it out the way that we typically see it in many texts, but there can be variations on the theme. Different intermediates may be cropping up if you look on different sources online. So what I would like you to know is the version that I'm going to draw for you right now. And you can remember the steps in tricarboxylic pathway with any of a number of different mnemonic devices. So I like to use, can I keep some sugar for my oatmeal? You can also, the one that's probably easy to remember is, can I keep selling sex for money, officer? Or one that a recent student of mine invented, which I like, is our class is, that's an alpha, ass, forget me. So you can either use the A or the K in alpha ketoglutarate. To generate your mnemonic device. So I also draw it kind of in this orientation because I remember where different outputs in from the citric acid cycle or the tricarboxylic acid cycle are found using an analogy of, it looks kind of like the face of a clock. Okay, so I'm going to start with oxaloacetate. And that's going to get converted into citrate. Now, oxaloacetate is a 4-carbon molecule. Citrate is a 6-carbon molecule. So we've got to add in 2 carbons. How are we going to do that? We are going to use what's left of pyruvate after pyruvate's already gone through an oxidation step. So those 2 carbons that are left after pyruvate oxidation form what's known as an acetyl group or acetyl. And that is linked to a coenzyme called coenzyme A. And coenzyme A is actually much, much larger. This is just, the acetyl group is just 2 carbons, plus a few other atoms attached to it. And coenzyme A is kind of a big honking molecule. So coenzyme A is going to dip out at this point, but it's going to attach those 2 carbons from that acetyl group onto citrate. So now we have that 6-carbon intermediate. And then that's going to get converted into isocitrate, which is an isomer of citrate. And you may see as an intermediate here a an intermediate that is called cis-aconitate. But I typically don't include that. But it's out there. You may see that it's in the textbook, but I typically don't include that in there. All right, so isocitrate is going to be converted to alpha ketoglutarate. And when we do that, two important things happen in this step. One is we harvest some energy releasing electrons in the form of NADH. We put those electrons onto NAD+, which is the electron bus, an electron shuttling molecule that can use those electrons at electron camp, or the electron transport chain. So of course, isocitrate, being an isomer of citrate, is going to also have 6 carbons, but as we pull those electrons off, we're also going to produce a molecule of carbon dioxide. And if we're accounting for carbons, which we should do, that's going to mean we're down to 5 carbons in alpha ketoglutarate. Alpha ketoglutarate is going to experience some changes. Coenzyme A, it's really just coming in there. It's not leaving just yet. Remember coenzyme A from up here? Well, it's back, baby. And coenzyme A is going to join to alpha ketoglutarate and form succinyl-CoA. But in that process, again, this is an energy harvesting step. So we're going to take some electrons, put them onto NAD+, the electron bus, send them off to electron camp. And we're going to remove a second molecule of carbon dioxide. So now we've gone from 5 carbons down to 4 carbons. And as you can see, once we get back to oxaloacetate, there's no more carbons that are going to be given up. All right, so the rest of this, we're accomplishing something other than oxidizing carbon. Then we get to our second S, which is succinate. How do you remember that succinate is our second S, not our first S? How do you keep succinyl-CoA and succinate in the correct order? Well, remember that succinate is late in this cycle, so it's the second S. And this step typically produces a molecule of a high energy nucleotide, like ATP. It can be ATP, but often it might be something else. It might be GTP. So in our text, it says this step produces GTP, whereas in previous versions of this course or previous biology courses, you may see that this 6 o'clock step here makes ATP. But GTP, just as good as ATP, just used for different things. Our next step is what I call the F step, because we're going to form fumarate. And as we form fumarate, we're also going to find, this is probably the only place we're going to see this occurring, a second different electron bus, or a third if we include NADPH. So this is flavin adenine dinucleotide, or FAD, which when it gets reduced, it's going to pick up 2 protons plus 2 electrons. So that's going to give you FADH2. This is considered to be a reduction, even though you're not changing the charge at all. You are picking up electrons with some protons, but this is still considered to be a reduction because FAD, that's its job. It is an electron shuttling molecule. It is an electron bus. So that's our F step there. Next step, fumarate is going to form malate. Keep track of our carbons here since we're doing that. And nothing too remarkable there. No major life-changing things, just isomerization. And our last step is going from malate to oxaloacetate. And here at this step, one more time, we're going to harvest some electrons. We're going to produce a third molecule, 1, 2, 3 molecules of NADH. So this cycle, it accomplishes a couple of different things. One is it produces energy. This is a catabolic process. It's releasing energy, converting energy into electron-rich molecules like NADH, FADH2, and we're getting at least a little bit of substrate level phosphorylation where we'll get ATP or GTP or something like that. And that's pretty special. Getting energy is a great thing for cells to do. They have high energy needs. So, yep, that would be reason enough. But the other thing that we're doing is we're doing a lot of isomerization, a lot of shuffling of atoms around in things like succinate, fumarate, malate, oxaloacetate. And these molecules are part of biosynthetic pathways. So they're going to get used to be made into other things like amino acids, coenzymes, and things of that sort. So that is, in a nutshell, why the tricarboxylic acid cycle is something that we need to know about because of its importance in biosynthesis and in energy harvesting. And maybe just for funsies, while I've got you here, why do we call it the tricarboxylic acid cycle? Well, I'm just going to draw one of these intermediates, citrate. Look, there's a carboxyl functional group. There's a second carboxyl functional group. 3 carboxyl functional groups. So I've drawn this as citric acid. So there's your 3 carboxyl groups, which is why it's the tricarboxylic acid cycle. Okay, and there are enzymes that catalyze all of these reactions, but I am not going to draw them in for you. Just know that all of these reactions are catalyzed by enzymes because that's how cells do things. Talk to you later, microbiologists.